Many years ago, my PhD advisor told me to succeed in science, find the lowest hanging fruit, and publish often. Starting my lab 3 years ago here at Stanford, I found perhaps the highest hanging fruit. Our goal is to try and 3D bioprint the whole human heart as a lab. It is extraordinarily difficult. In fact, harder than perhaps I expected many years ago it might end up being. We focus our lab on this particular condition, hypoplastic left heart syndrome. It's a congenital heart defect where instead of being born with 2 functional ventricles, like most of us in this room, you have 1 functional ventricle. And as a result of this, deoxygenated blood coming back from the body that expects to be sent to the lungs ends up mixing with the oxygenated blood coming back from the lungs and expects to be going to the body. And that mixed blood is not enough oxygen to keep that baby alive beyond 1 or 2 weeks of life. Now, thankfully, back in the 70s, an Italian physician, Dr. Fontan, discovered this amazing surgery that allows you to connect the body in series with the lungs, so that instead of visiting your heart twice per circulation, now the blood only goes once, and the deoxygenated blood from your body goes straight to the lungs before revisiting the heart. It's really a miraculous procedure. However, it only offers about 20 to 30 years of median survival. That's enough if you perhaps need a heart transplant at 70, but these are children, 2 years old. These are children that will have limited exercise capacity, and by the time they're 20 or 30, will need either a heart transplant or succumb to their illness. So we dream of changing this little pipe that they put in to connect that in series that's passive, that sits there, it's just a pipe, with something that's living, that's beating, that's made up of the patient's own cells and has valves to enable unidirectional flow. This is an extraordinarily difficult goal. It would need to have thick walls to achieve enough pressure to work like a second ventricle. It would need about 3 billion cells. Anyone that does stem cell culture is used to working with cells in the millions, certainly not billions. It needs blood vessels to keep all of those cells fed with the oxygen and nutrients they need to survive, and valves to make sure that the blood flows forward and not backwards. And manufacturing tissues like the heart, liver, or kidney is extraordinarily challenging for several reasons. First of all, there's a lot of cells. Compared with tissues like cartilage or tendon that have made significant progress in being able to tissue engineer, solid organs are incredibly cellularly dense with many cell types crammed at very high densities. And these cells are organized from the centimeters down to the submicron scale. There's incredible complexity across all of these orders of magnitude. And most importantly, and in fact what's really been field defining and limiting for tissue engineering has been the incorporation of a pervasive vascular network that's able to feed all of those cells, all of those nutrients and oxygen molecules that they need to survive. Now, thankfully, there's real reason for hope in the field of tissue engineering. Some of you may know we can now create your own pluripotent stem cells. That's an embryonic-like stem cell. And from that, we can create a whole myriad of different cell types from your own cells. This is an incredible cell source and opportunity for tissue engineering. Many other labs around the world have perfected the way of instructing these cells to self-assemble into miniature tissues that are able to recapitulate a lot of the functions on the small scale of organs. And finally, this really cool method of 3D bioprinting called 3D embedded bioprinting, where you're able to write one soft material inside a second support material, really unlocks our ability to write not hard plastics like many of you have seen with 3D printing, but stem cells. These are soft, gooey, biological materials, right? Not the nice engineering polymers we're used to working with as engineers. Biology is gooey, and it needs to be supported while you're printing it. If you try and print mayonnaise in that sort of tree-like shape, in air, it's going to flop over, okay? So this embedded 3D printing allows us to recreate complex 3-dimensional networks. So our laboratory here at Stanford, now 3 years old, is embarking on this holistic pipeline of making billions of cells from stem cells, rendering them into mayonnaise-like inks that we can print. We can introduce vasculature, and for the first time really keep these cell-dense tissues alive. And that's the first step in now understanding how to get the most function. We work on making lots and lots of these microtissues using the latest approaches in synthetic biology. We combine hundreds of thousands of these to then write channels into the tissue with a 3D printer that we can use to perfuse those tissues with oxygen. We're working on scaling this up so that we're not working with little bits of tissue in a petri dish, but really want to get to that liter, kilogram scale of tissue and ask, how do these behave at that scale? And looking really down at the science of printing these mayonnaise-like inks comprised of thousands of human stem cells. I'm going to focus just on 2 of these stories a little closer. So these little pieces of tissue are each about 2,000 stem cells that have been instructed to become cardiomyocytes, that's heart cells. And you can see them there in green, the heart cells that express the markers that tell them to beat. And indeed, when you look under the microscope, you can see these little things jiggle. It's super cute and super exciting. I never get tired of seeing these things. Now that is our mayonnaise-like material that we are going to use to build a bigger tissue. And that poses a real problem. If I combine those together, are they going to beat independently, or are they going to work as one and beat synchronously to be able to form a larger tissue? And by day 1, they don't really talk to each other. But by day 10, we figured out a cocktail that allows them to fuse together and start beating much more synchronously like a heart would. So we can put our printing into these mayonnaise-like slurries of all those aggregates. If you look carefully, you can just about make out those little dots, but now there's hundreds of thousands of them. We can print channels, evacuate material, perfuse blue oxygenated media through for visualization, and after 16 days of culture, here we have a centimeter-tall wodge of heart tissue beating and pulling on some springs. So this shows that we really can start to think about scaling up cell production and perfusion. And these structures, they go right from the centimeter scale down to the submicron scale, where we can see sarcomeric alignment, these little motors that are doing that beating. We can direct how they beat. And we can test drugs on these tissues. So what I showed you was, many, many hours of late night working in lab to get enough cells for one print. And reviewer number 3 in this paper said, I think very reasonably, that's a lot of work for making a Y-shaped channel. We do not want to stop at making a Y-shaped channel. We're working with Alison Marston here at Stanford, who's an expert in hemodynamics, who's able to automatically design a vascular network for us that is ideal for perfusing any organ shape we give it. And Jesse Herman, a very talented medical school student, has been working in our laboratory to figure out an algorithm on the order in which you have to print all these vessels so that you don't pass through a previously printed vessel, which would break it. And so this really speaks to the need to scale up our production. We can no longer work in petri dishes. We need to work in much larger bioreactors. And so this is the industrial heart of our lab, where Stacy Lee, a research scientist with us, is opening up one of the reactors so we can get a closer look. And every single one of those little white pieces is not an air bubble. Those are little aggregates of heart tissue that are able to beat. And when we compile those together, that's a 20 milliliter pellet of cells. That is a vast, vast quantity of cells in a single batch. And with this, that's about the number of cells that we need to start printing this entire human biopump that I showed earlier. So we're very excited to begin working at this much, much larger scale with our production. And we recently got supported by, this is Biden's latest agency, ARPA-H. Some of you may have heard of DARPA. There is now an ARPA for healthcare. And we were one of the 6 awardees of the first inaugural year of ARPA-H to bioprint the entire human heart and put it into a large pig model within the next 5 years. So I don't expect any weekends for about 5 years. But it brings together a great set of investigators and collaborators that we can all work together, and this can only be done at Stanford. Stanford, I challenge you to think of any other university with a world-class medical school right next door to a world-class engineering school. There are none. This is the only place we can do this kind of multidisciplinary work. So with that, I really want to thank the people that really sit in the lab and do all of that culture to make all of these dreams possible. And I'm happy to pass it to the next speaker. Thank you.